Hangin at the Reef is brought to you by Hawaii Pacific University's Accelerated MBA, a 12-month program featuring access to certifications from Google, Meta, and other leading companies. Courses are offered online, in person, or in hybrid formats. Learn more at hpu.edu/MBA.
Our guest today is Dr. F. David Horgen, a professor of chemistry at Hawaii Pacific University, where he also serves as chair of the Dive Control Board, head of the Shared Instrumentation Facility, and faculty athletics representative. As the principal investigator of the Laboratory of Marine Biological Chemistry, Dr. Horgen's work is at the forefront of marine natural products research, exploring how compounds from the ocean's diverse ecosystems can lead to medical breakthroughs. With a PhD in pharmacognosy from the University of Illinois at Chicago and a postdoctoral fellowship in marine natural products chemistry at the University of Hawaii in Manoa, Dr. Horgen's expertise spans organic chemistry, biochemistry, and bioanalytical chemistry. His research focuses on uncovering marine-derived compounds with potential applications in treating diseases like cancer, viral infections, and neurological disorders. His work has earned him numerous awards, including the HPU Teacher of the Year Award, the Weinman Innovator Award for Translational Research, and the Jack L. Beal Award from the American Chemistry Society. Today, we'll be talking about his cutting-edge research on antiviral compounds, marine natural products, and the incredible potential that ocean-derived molecules have in modern medicine. We'll also explore the role of students in hands-on scientific research and the exciting future of marine-based drug discovery. Let's welcome Dr. David Horgen to the podcast. So David, your research is truly cutting edge. You are at the forefront of research and uncovering compounds that can cure all sorts of diseases and ailments. And aside from that being a very fulfilling career to have that much of an impact on health among people, you've got just interesting things going on. And I wanted to start our conversation by asking you about the HPU Natural Products Library and hoping you can explain that to our listeners and tell us a little bit about what that is and how your work is involved in that library.
Yeah, so I think probably the first piece of that is it's a library, but I'm a chemist. And so a library to a chemist is really a collection of chemicals. And so we are interested in finding chemicals from nature that have biomedical potential. And so one of the nice things about living organisms is that we produce really interesting chemistry. And the types of chemicals that we produce, our bodies are made up of chemicals. We're all chemical, every piece of our being. But these chemicals that we produce, some of them have functions that are very similar to drugs. And in fact, about half the drugs that we use in modern society, they derive from naturally occurring compounds. And we use natural compounds like Taxol, like vincristine, even things like penicillin and opiates and many other compounds, hundreds of them, and types of compounds that we use that are directly from nature. But then there's a whole class of compounds that we use as drugs that are based on natural compounds, slightly modified. And so you start with the naturally occurring compound and you change it a little bit just to make it a little bit better. And then we also have compounds that are based on structure. They're made in the lab, but they're based on the structure of naturally occurring molecules. So about half the molecules that we use for drugs have their basis in naturally occurring compounds. All right, what's HPU Natural Products Library? It is a collection of extracts from marine organisms collected in Hawaii, where we put them in a format. We have them sort of and we call them plates, but they're in little test wells, little tiny test tubes, basically. And we have these extracts. We make extracts kind of like you make a coffee or a tea, and we use different types of solvents in order to make these extracts. We might use things like methanol that people might have heard about, and things like hexane, which is related to some of the chemicals that are used in our fuels and at the gas pump. But we make these extracts, and now we have these collection of chemicals from organisms, and we test them, usually in collaboration with disease biologists, for activity that will either play a role in preventing or treating a disease. So the HPU Natural Products Library is a collection of extracts from organisms that live in the ocean, mainly plants, invertebrate animals, and microorganisms.
So are those all single molecules or not compounds yet? So you've got essentially isolated ingredients in the library, and then you're figuring out how to combine some of those into compounds that then cure disease and do other things.
Well, it's actually kind of the other way around. So our libraries are actually collections. They're complex mixtures of compounds. So if we test a complex mixture, we find a biological activity that could be helpful or useful in disease, then we take that collection and we isolate from that collection what are the molecules that are actually causing the effect that we want. And so we've done this, over and over again. We found compounds that have a potential role in the treatment of stroke, cancers, different types of cancers, and we're also now recently working on anti-COVID compounds.
So tell us how these natural compounds are screened for in biological activity. What's that process look like?
Yeah, so there's a term called high throughput screening. So the main way that drugs have been developed is through a process where we call it screening, sometimes called high throughput screening, where we have a biological test that can be run at a very, very, very small scale, and we can run many tests all at one time. And the test is a biological phenomena that we're testing for. And so first of all, you have to design that test. And so we've done some of that, and we focused on proteins that are in our cells called ion channels. And we look for ways to turn the ion channel on and turn the ion channel off using these collection of compounds, the HPU Natural Products Library, using the collection of compounds. So, for instance, we had, there's one channel called the TRPM7 ion channel, and we screened about 14 or 1,500 different extracts that are in our HPU Natural Products Library. And we found a handful, I think about 8 of those 1,500 samples that caused the effect that we thought was important and from a biological perspective. So we're doing this in the lab, where we're usually it's cell based, so we grow cells in little tiny test tubes. We call them plates, but they're little tubes on these little flat plates. And we do the tests. And by having the biological event, and we're interested in turning off and turning on the protein, and so we can measure that, and then we subject the proteins in the cells that have the proteins to the extracts. And if you add the extract, and then the effect that you measure is that the channel was turned off, then you go, that's what I like. Like, okay, that is a hit. We call them hits. And so that becomes then something that we look at in more detail. So that's the first step, that was finding a hit, an extract that has the biological effect that we think will translate into either treating or preventing a disease, or in some cases, understanding diseases better.
Wow. So it sounds like this step in the research process is in the lab and looking at reactions of chemical compounds. How far down the road does this have to go to get to the point where you're testing in live subjects, for example?
Yep. Years. I mean, it takes an average of about 15 to 20 years to develop a pharmaceutical, to develop a drug. So the probably 10 years to ever come close to a human being in many cases. And our pace of research, we do lots of different things. So our pace is not like the pace in a pharmaceutical environment. We go at a fairly low pace, but once we reach a certain point, then momentum can really build pretty quickly. And we're kind of at that point with one of our compounds where the momentum is building pretty quick towards, we're putting it in animals, and we have collaborators that specialize in animal studies, and so that could be in human beings probably within 5 years if everything went beautifully.
I know one of the projects that you had mentioned that sounds really interesting is you're working on the development of a lead drug for hypoxic events in newborns. Tell us a bit
Yep.
about how that process has evolved from identifying the molecules that could create, effective compounds to where you are now in that research.
Yep. So, okay, we exactly found, so there's this compound called, well, it's a collection of compounds, but the main compound is called boyzinosin A, and it's from a soft coral that grows here in Hawaii. And we found the molecule. Now, the molecule had been described before, but we've also found other new compounds that were closely related to it as well. But the activity of it, we discovered just exactly as I described. It was in our HPU Natural Products Library, an extract of the soft coral, and we tested it and we found a hit. And it was one of the more attractive hits, just based on it how potent it was. And we were able to isolate the compound. And then we worked with collaborators. We have collaborators all over the world that are working on this compound actually, but our main collaborators are at the Queen's Medical Center, at Baylor University, and at University of Toronto. And so we worked as a team. It took us about 4 years, and we worked to get funding from National Institutes of Health, from the National Institutes of Stroke and Neurological Disease. And we had funding that goes on for about another year to develop boyzinosin A as a treatment for hypoxic ischemic brain injury. Now, that is what happens in newborn babies if they have if there's an event that prevents them from getting enough oxygen. And the protein that boyzinosin A inhibits or turns off is overactive and kind of goes out of control in brain cells when the brain is starved of oxygen. And so we have a very plausible mechanism of how this molecule can positively impact this state in newborn babies. There is no current treatment for this if it occurs, if the event occurs during birth or prior to birth. There's the only treatment is to reduce the temperature of the newborn, or of their just either the whole body or their brain. For about 72 hours, you lower their body temperature, and that helps to reduce the damage, but it's not nearly effective enough. So our goal is to create a drug that can be administered to newborn babies that meet a certain criteria where they've had an event in order to prevent brain damage. And so when this does happen to newborns, they have, depending on the severity, it can lead to just a lifetime of debilitation, and all the way from very mild to very severe brain injury has impacts throughout life.
So how do you identify or pick, the ailments that you're going to address? So for example, for this research, did you stumble across the molecules and the compounds and then determine that it could have an effect on this particular illness? Or were you working specifically on identifying compounds that could solve that illness in newborns?
That's a very good question. So I come at it from the perspective of that nature is full of complex, highly diverse chemical structures, and that mimic or have the correct types of properties that can serve as drugs. And to serve as drug, a molecule has to be able to bind to a protein. So I come at it from that there is potential there. And then I work with biologists to find what are the most relevant, most, sort of cutting edge areas that we can look at. And so back in, we started working with the TRPM7 channel back in around 2006 in collaboration with Reinhold Pinner and Andrea Flag over at the Queen's Medical Center. And they were the team that discovered this channel. There's 3 teams in the world. They simultaneously discovered this protein, the TRPM. And so we started working with them. And because we saw the potential initially as a treatment for cancer, we thought initially that was perhaps one of its most important properties. But in the meantime, other biologists all around the world have found that this channel, it's involved in malaria. When the organism that causes malaria gets into blood cells that protein plays the tryptophan 7 channel, TRPM7 plays a role. We found that it plays a role in cystic fibrosis. We found that it's not, I mean, in the literature and with my collaborators now that I have a network around the world providing this compound to them. And then we also then kind of recently, the involvement of this channel in the metastasis of breast cancer has become a very important, hypothesis in building new treatments for breast cancer. And so we are working actively with Jason Lee over at the Cancer Center. He is the expert. He is the expert on the disease, and we are the experts on the chemistry and on how to pull out and figure out which of the compounds are causing an activity. So this compound Wyzinsin A, we're now looking at as a potential treatment for breast cancer as well. So that wasn't a very clear answer, perhaps, because it's kind of, I would say it's a little bit that what disease that we work with depends on what's known and what's being discovered, and finding outstanding disease biologists to work with.
So I imagine research into drugs to treat cancer is a big part of your overall research. And you just mentioned one example. I think you had talked about another cancer research initiative involving terrestrial plant extracts and a collaboration with the Honolulu Botanical Gardens. And I think that was addressing targeting non-small cell lung cancers. Can you tell us a little bit about that research and where you're at with it?
Yep. And so that's actually also in collaboration with Jason Lee over at the Cancer Center. He has another area of expertise. And so the Honolulu Botanical Gardens collaborations is kind of an interesting thing. So I did my graduate work at University of Illinois at Chicago, and I worked on terrestrial plants. I trained as a botanist, and a piece of my training was in botany. Mainly, I trained as a chemist and a pharmacologist. And then when I came to Hawaii to work with Paul Scheuer, originally at University of Hawaii, before I joined HPU, I shifted my attention completely to the marine environment. And so the Honolulu Botanical Gardens in the last 3 to 4 years has been sort of a shift for me back to the terrestrial environment and organisms that live on land. And so I've been working with Naomi Hoffman over at the Honolulu Botanical Gardens, and we've been in collaboration building libraries, making extracts, developing extract libraries from their collections. And they have plants from all over the world, and it's a very organized gardens and lots of information about all their plants. And so they've been wonderful to collaborate with. And so we are taking this new developing collection of extracts from plants that have been sourced at the Honolulu Botanical Gardens, and we are going to screen them. I said high throughput screening. We're going to screen these extracts for their ability to inhibit the growth of non-small cell lung cancers. And we have a bit of a strategy there. And it's a sophisticated strategy that it's like by myself, I would not be able to fully implement. So it's really these collaborations that we build that give us really enrich sort of the sophistication of the work that we're able to do here at HPU, and to leverage our expertise and our resources. And we engage our students really in a sort of across a much broader experience and tool set they get to develop.
Yep. Well, I want to come back to, hands-on research and how students get involved in all of this in a minute, but I want to stick with, sort of your sources for molecule identification. So you've talked about unlocking secrets from coral, from plants. Where else can you go to find these? Recurring, and we've heard about medical products being derived from snake venom, for example. Is it really limitless? And the applications are clearly unknown, but I imagine there are tons of other sources to go find these molecules.
Yeah, there are, and I think sometimes students have great ideas about what they want to do. But I'll give you an idea of some of the things we've done. So we've worked on organisms that live in the ocean. But one, you mentioned a toxin. One of the toxins we've worked on is from box jellyfish. And, box jellyfish They come in after the full moon and they cause some trouble. They don't feel good when you get a sting. They're actually beautiful animals when you see them swimming in the water. But then we studied that with a collaborator over at University of Hawaii, who's a venom specialist. We studied box jellyfish venom, looking for bioactive compounds, and we published on that just a couple of years ago in the journal Molecules. And I have a current collaboration with J.P. Bingham, Professor Bingham, also at University of Hawaii, and he is a cone snail expert. So we're working with him and his students, mainly his students, come over and we just have this very great kind of open, collaborative environment in the state of Hawaii, as you can kind of imagine. And so, yes, and snake venoms, well, we don't have snakes in Hawaii, so there's no snake venom expert, but there's...
That's true. I forgot that. No snakes in Hawaii.
Yes.
No snakes, no squirrels, and no dodo birds, I guess, right?
And the one limitation is that some like vertebrates that we're not that great at chemistry. And so vertebrates like mammals and fish, we don't do a lot of like really, really cool chemistry like plants do, and like invertebrate animals like corals and sea cucumbers and idarians like the jellyfish or like microbes, like blue-green algae, bacteria, and fungi. These organisms produce a lot more in terms of the diversity and the number of interesting molecules that could potentially serve as drugs. So we're not going out and collecting fish in order to extract and then find interesting chemicals. We do extract fish to find ciguatoxin. So we do have another whole project on looking at ciguatoxin, which is food poisoning. And so using some of the same tools that we use to measure compounds, we measure that in fish. But we're not extracting, we're not studying fish for their biomedical potential, but we'll study the algae and the microorganisms that they eat and small little animals that they eat because they produce interesting chemistry.
Wow. So, you're working on the development of drugs that can treat illnesses after they've occurred, but as anybody in the healthcare system would say, the holy grail is, of course, preventative treatments to keep disease from occurring and to solve, imbalances in our body's chemistry, for example, that lead to other chronic and acute illness. So I understand you're also doing work in research into food and nutrition for disease prevention. Tell us a little bit about that research and where you're at with some of those initiatives.
Yeah. So, okay, there we have one project. So this is maybe more about interest and less of a mature sort of domain where I'm working in. But one of the things that we did do was work with a investigator in Montreal who's a specialist in food preservation and food microorganisms and food poisoning. And so we had actually used our extracts to look for compounds that could then be used to prevent food-related, foodborne disease. So that's one area, right, where diet and the intersection with these bioactive compounds. But at the same time, and we're doing that with the ciguatoxin, looking at diet of fish consumption and the risk of food poisoning from ciguatoxin, which is produced by little small photosynthetic organisms. But the other area, I think, where there's some growing need and interest, I think is very appropriate for Hawaii, is looking at diet, understanding dietary components and how they impact risk for disease. And one of the challenging parts of that is that nutrition research it's very difficult to get very firm, clear answers about nutrition. And the reason is to gather that information, to really know something about health and health impacts, you have to have large trials because there's lots of variability in the human condition and in our biochemistry. And so in order to really understand, like, okay, if you eat lots of fruits and vegetables, does that impact your incidence of cancer? Well, that's a hard question to answer because you need long time periods before you can understand, did people get cancer or not? And then you have to have good information about, well, what is their diet? And these are all challenging things to collect. So I think looking at diet, looking at prevention is, I think, an important arena. And there are ways to do it that to get answers in a much shorter period of time by, again, working with biologists that specialize in the prevention of disease and finding molecules that are known to intervene in the biochemistry that short circuits maybe the production of cancer-causing compounds in the bodies. There's a enzyme called quinone reductase that, when it's elevated in concentration, it actually reduces harmful effects of cancer-causing compounds or chemicals that we might be exposed to. So I think that that's an area in Hawaii where there's a lot of room for growth, is looking at prevention of disease, looking at diet and dietary components. How are we feeding ourselves? Where is the food coming from? And what specific items of food are we eating? I think it intersects with the idea of food security. We import a lot of our food. As we increase our food production in Hawaii, how is that going to impact the nutritional content of our diet? I think there's a lot of interesting questions to be looking at, and I'm curious and I'm interested. But that's an area that I think it's a future area of research for us.
Yeah, for sure. And I love the idea of moving the focus to prevention versus treatment of illness. And, I think, as you said earlier, we're made up of chemicals. Our bodies are chemical compositions. And if you believe that certain chemical imbalances lead to illness, whether physical illness, whether mental illness, and the balancing of your body's chemistry can help solve those problems. But it seems like our medical system is focused on the treatment of acute illness after you've had an episode or you're already suffering from a mental or a physical ailment. Is there a belief that in preventative medicine that these compounds that are being created really are the panacea for balancing our body's chemistry to create the wellness factors that prevent these illnesses down the road? And that with more focus on understanding your body's chemical composition, where perhaps your deficiencies may be, and understanding that and then supplementing that chemistry to balance those things out is perhaps the best pathway to long-term good health and the prevention of acute illness and long-term chronic illness.
Yeah, I mean, I think those are all valid points, and I do think I think those are whole areas of research that are perhaps underoccupied, within our biomedical research sort of nationwide community, right? And I think we would do a service by helping to expand sort of our understanding in those areas to better understand how nutrition impacts us. I do think, though, that part of our impact, and I know you may have some thoughts on or interest in how this impacts students, but many of our students that we work with in the lab are bound for med school. Many of them are bound for research, but many of them are bound for med school. And it's really important that they get a experience and understanding of research as they begin their medical training. It enhances, how the level to which they understand the information that they're using, how they use that information. And I think just being skeptical, maybe curious is the better term, but being curious and being open is one of the things I think that participating in the research really does enhance and opens our eyes. So I think an indirect way that we contribute to sort of the improvement of, our modern medical approaches is by ensuring that we're engaging our undergraduate students in rich and meaningful research experiences. So they kind of get that spark of curiosity and desire to help make and develop best
Yeah, well, it's certainly encouraging to hear that more med school bound students are getting exposure to research and the impact of preventative treatments as opposed to, again, just the treatment of the acute illness. I've heard that complaint about, the medical profession for many years, that there is not a focus on nutrition, and it's all about treating, these ailments as they come along. So that's really encouraging to hear. You touched upon how students get involved in this research. And I think that we would say here at Hawaii Pacific University that our programs attempt to integrate as much as possible experiential learning and hands-on involvement in research in particular. So give our listeners a taste of how HPU students truly do get involved in these projects.
Yeah. We have undergraduate programs, and we have a very nice master's program as well. And I would say the large bulk of the biomedical research that we do here at HPU is performed by undergraduate students. I mean, if an undergraduate student gets involved in research, which is our focus in most of our programs, they are going to be doing the most important work. They're not going to be just supporting someone else and doing research. They're going to be doing the research. I do have a picture that I show to students of me out in the field. Actually, I have a picture of me underwater, and I say, here's I'd like to show this to make my colleagues jealous of me, but I really don't spend any time in the water to speak of. I spend a lot more time in the lab showing students teaching students how to do science, and then I have a picture of me in the lab with students. And I said, but I really don't spend most of my time there either. I spend most of my time, I have another picture, it's me sitting right here, and I'm sitting on a bouncy ball right here, and I've got my computer screen in front of me, and I've got a coffee mug, right over here. I say, but I'm a pretty active researcher. How's the research being done if I'm sitting behind my desk? And because I'm doing all the stuff it takes to create the opportunities for research. And so then I have the next picture is students working in the lab. That's how the research gets done. They do the work. They learn to operate highly sophisticated machines, to do highly sophisticated projects. And then they come back and they sit in front of, and on the other side of the desk with me, and I have dual screens, and we look at the data, and we talk about the data, and we process the data. And the only time I get to go in the lab is when things break. And so I kind of look forward to when our mass spectrometer breaks, because I get to go over and try to do the first round of troubleshooting and try to fix it. Yeah. So we have 40 or 50 students a year that are participating in undergraduate research, and they just blossom. It's really fun to watch. And for many of them, it's life-changing. It's highly valuable or life-changing.
Wow. Well, it sounds like an amazing experience. And I'm sure that, our students get, as you mentioned, tons of time in the lab, but is being out in the field, on the research vessel, for example, exploring coral and plants in their natural environments, a big part of their work here?
Yeah, I think it depends on the research group. But yes, we have students that are out on one of our projects where we're looking at the ciguatoxins in fish tissue. Well, first, the ciguatoxin settles on algae. And so students are out collecting algae in the water and performing separations and isolating the components that we need to investigate. And so they're doing that on a weekly basis. And we've got other students that are working in traditional farming environments, looking at, taking water samples or getting collections of fish and invertebrates to study different aspects of those systems.
Very cool. Well, what can we expect in terms of, future studies and collaborations? Is there something coming down the pike, for example, that you're really excited about? But give us a taste of what's coming.
Well, I already mentioned one, and that is we're working on breast cancer right now. And so I think the most important factor for how likely some breast cancers are to spread is the level of the protein I told you about, the TRPM7. If you measure in the tumor how much TRPM7 it has, the higher the level, the more likelihood that type of tumor is going to spread. And that is what kills people that do have breast cancer or that have cancer in general. It's the spread of the cancer that really causes the the people to succumb. And so I'm excited about that. Like we have a molecule that has some real potential to impact a new way of treating breast cancer. So it's not done that way at this point. And then we have another project where we're looking at cannabinoids. We have it's through the National Center for Alternative and Complementary Medicine, and we're looking at the pain relieving properties of molecules from cannabis. And so that's a new project that's just about to get started in collaboration with Oceanit, which is a company that is one of our neighbors here down in Honolulu.
Well, that's exciting to hear that, with some of the renewed views about cannabis products and their efficacy and the healthcare equation, that the medical field is now starting to turn their attention to researching more intensely, the treatments that can be derived from those products, which I have to believe are going to lead to a lot of breakthroughs in pain management, mental, treating mental illness in certain ways and balancing, issues around depression. So that's really exciting and positive to hear. I understand too that on a personal level, that you have a connection to natural products, gardening and farming. So this sounds like a labor of love for you in many ways.
Yeah. So I am a farmer. And so I am funded by the USDA to have a mixed agroforest. Maybe I'm more of a forester. And I have my main crops are breadfruit and cacao. Now, cacao is what produces the fruit where we get chocolate from. Chocolate is a incredibly rich source of antioxidants, of cell protecting molecules. I drink, you can see right here, drink cacao. I drink cacao. You just take a spoon of this and you put it in hot water. This is just regular cacao from unsweetened. And so you don't want to add the sugar. You don't want to add the milk, just the cacao.
It's not going to taste like chocolate, right?
But it's kind of like drinking black coffee. You come to really enjoy it. But anyway, but cacao and then the breadfruit. And so breadfruit is an amazing plant. It's a canoe plant that was brought by the Polynesians that migrated to Hawaii to create the great Hawaiian civilization. And breadfruit is an amazing plant, and it's a very nutritious source of carbohydrates and a little bit of protein. And so I'm interested in the nutritional aspects of certainly of breadfruit and of cacao and other products of Hawaii. I mean, coffee is a really important product in Hawaii as well. And so I do have a lot of sort of vested interest in nutrition, in sustainable agriculture, and also food security. And so I have a personal ethic of trying to maximize the amount of food that I eat that is harvested in Hawaii, whether it's meat products like fish or game, or vegetable and fruit products as well.
Wow. Well, I know the history of cacao goes way, way back as the drink of royalty in ancient times, for example. And I would imagine that there was an understanding even back then of some of the health benefits that come from consuming that particular product. Well, David, this has been a fascinating conversation, and we're really excited to have you back and to hear more about some of the outcomes of this research and also some of the new initiatives that I'm sure you'll be working on in the future. But, can you tell our listeners where they could go to learn more about your work or to connect and learn about some of these fascinating research projects?
Yeah, well, I think that's a great question. We do have a website, and that gives us information about our program. And we have, most of the information that I produce is out. It's on the internet in the form of scientific papers. So if you go to PubMed, you can search for our work. And I think you've done some stories on some of our work too on our in our HPU.
The Ohana published articles. Yes, that's a great source for information on some of your research projects. We've written up a number of feature stories about your work.
And we mentioned the work, and what I'm probably most proud of, though, is the students that we do send off into the world well prepared, either to pursue careers in medicine, which includes dentistry and MDs and DOs and pharmacy, And then in research, we have just this year, a student is starting at the MIT PhD program in chemistry. We've got a student at Oregon State that's doing amazing things in their PhD program. We've got other students accepted Washington State, Baylor University, PhD programs in chemistry. And so we have a really nice pipeline for our students to really advance in their careers and in their education and their careers.
Fantastic. Well, as I said, I'm excited to have you back again on a future episode. And I just want to say thank you for a fascinating conversation and enlightening all of us on all of the amazing research opportunities that are happening here at HPU and I imagine across other schools to really solve some of the longer term medical problems. And I think the research into more obscure compounds coming from coral, animal byproducts, plants, is just such a fascinating and impactful thing to be working on because the benefits are substantial for human health, advancement of medical treatment. So it's got to feel good to be at the forefront of that and to be teaching and leading students who are going to carry that mission forward as well. So I want to say thank you for your time today and say I'm excited to have you back and to talk more in a future episode about your research.
Thank you. I enjoyed it and look forward to coming back. Aloha.
Aloha.
Hangin at the Reef is brought to you by Hawai'i Pacific University's Accelerated MBA, a 12-month program featuring access to certifications from Google, Meta, and other leading companies. Courses are offered online, in person, or in hybrid formats. Learn more at hpu.edu/MBA.